Hello dear students. In this video we are going to study about the giant cells. In our previous video we have studied about the granuloma and the types of granuloma. So in this video we will see how the giant cells, different type of giant cells look. Okay. So there are two types of giant cells. The first one is the physiological giant cell and the second one is the pathological giant cells. So physiological giant cells are found normally in our body. So where do we see these giant cells and what are the different types of physiological giant cells? We will see it. Okay. The first one is the syncytiotrophoblast. The second one is the osteoclast and the third one is the megakaryocytes. Okay. The syncytiotrophoblast we can see in placenta. In the normal condition we can see syncytiotrophoblast, and osteoclasts are seen in bones. We know osteoclasts are the bone resorption cells, and the third one is the megakaryocyte, which is found in bone marrows. And we know that megakaryocytes are the precursors for platelet. Okay. Now the pathological giant cells. We know that the giant cells are the multinucleated cells. So there are different types of pathological giant cells which are seen in different types of diseases. So the first one is the Langhans giant cells. In case of Langhans giant cells, the nuclei are arranged peripherally in horseshoe pattern. Okay. These cells, this schematic diagram shows the Langhans giant cell, and these round cells which are arranged peripherally, these are the multinucleated cells. These are the nuclei and these are arranged peripherally. So this is forming a pattern which is similar to horseshoe pattern. Okay, that is why it is called the horseshoe pattern, which is seen in tuberculosis. Now the next one is the Touton giant cell. The nuclei are arranged in ring. You can see that nucleus is arranged in a ring form surrounding a central foamy cytoplasm. Here you can see that this whitish area is the foamy area. This whole whitish area is the foamy cytoplasm. So this is seen in case of any fat necrosis. Okay. Now the next one is the foreign body giant cells. So in case of foreign body giant cells, the nuclei are arranged in haphazard. We can see that this one is a giant cell and the nuclei are arranged, this dot are the nucleus. These are arranged in haphazard pattern. Okay. This is arranged in a haphazard pattern. So the foreign body giant cells are seen in case of sutures or talc material if left inside the body for longer period. Now the next one is the Werthin-Finkeldy giant cell. These are the multinucleated giant cells with eosinophilic inclusion bodies in lymphoid tissues, which is seen in case of measles. In case of measles infection, we can see birth in Finkeldy giant cells in lymphoid tissues. The next one is the herpes simplex infection. In case of herpes simplex infection, we can see a giant cell, this one, which has 3 features, which we can memorize it by 3M. What are 3M? This is multinucleation, margination and molding. Multinucleation means there are multiple nuclei. Margination means all the margins of the cells are well demarcated. You can see the margins are well prominent. Okay. And molding, that means all the cells are trying to fit into each other. So this one is seen in herpes simplex virus infection. The next one is the Reed-Sternberg cells, which is seen in case of Hodgkin's lymphoma. Hodgkin's lymphoma. How does it look? It looks like a binucleate owl-eye appearance. This one. This cell is the giant cell, the Reed-Sternberg cell, and there are 2 nuclei which gives appearance of an owl eye. Now the last one is the Ashkoff bodies. Ashkoff bodies giant cells. These are the vesicular and ovoid nucleus with prominent nucleoli. Okay. So Ashkoff bodies giant cells are seen in case of rheumatic heart disease. Rheumatic heart disease. So this was all about the giant cells. Thank you.